Welcome to the segment on lipids and nucleic acids. This is all part of Chapter 5 in the Campbell Biology textbook. All right, so let's continue. So remember the macromolecules, they're large and they're usually complex, and what makes them complex is the various combinations of functional groups that may be attached to the skeleton of each one. And remember, all of these are organic molecules, so they are made up of carbon and hydrogen, plus some other atoms depending on what type of macromolecule we're dealing with. So remember, there are four classes of large biological macromolecules. We have carbohydrates, lipids, proteins, and nucleic acids, and each of them have unique properties that arise from the arrangement of their atoms and, of course, the number and location and type of functional group attached to the skeleton. So again, here we're going to be dealing with lipids and nucleic acids. So let's start with lipids. Lipids are quite a diverse group of hydrophobic molecules. Hydrophobic molecules are molecules that do not readily dissolve in water. So they're afraid of water. You can kind of describe them that way. Phobic means afraid of, and then hydro related to water. Now they don't readily dissolve in water because they're made up of mainly hydrocarbons, which is really just carbon and hydrogen. So they don't have any or many electronegative atoms within them. If they do, the ratio of hydrogen and carbon is a lot higher to those electronegative atoms that it makes them less readily able to dissolve in water. Now, lipids are the one class of macromolecules that do not include true polymers. They do not form polymers. However, some of them form by the same type of dehydration reaction in which polymers form in carbohydrates, in nucleic acids, and in proteins. However, they're not called polymers because they're just components and they're not just repeating units. So again, they mix poorly, if at all, with water, so they don't readily dissolve in water. And they consist mostly of hydrocarbon regions. The most biologically important lipids include the fats, the phospholipids, and steroids. So we're going to look at the structure and some key characteristics for. So if we look at fats, this is the one that I was talking to you about. Fats are formed by that same dehydration reaction that occurs in the macromolecules that actually form polymers. However, these fats do not form polymers. They just have components that make up the fat. So fats are constructed from two types of molecules, which are smaller, and then when they come together using that dehydration reaction, which is what we see here, they form a fat. So the two types of molecules are one, glycerol and fatty acids. So the glycerol is shown here. It's a 3-carbon molecule, and each of those 3 carbons have a hydroxyl group attached to them. So each of those hydroxyl groups can potentially react with a carboxylic acid found in the fatty acids. So fatty acids are called fatty acids because they're fatty, right? So they have this hydrocarbon region, and they're usually very long chains of hydrocarbons, often in even numbers, especially in living systems like humans. And at one end is a carboxylic acid. So this end right here would be considered hydrophilic, and then the end with the carbon and hydrogen only would be the hydrophobic region. And you can see the region that's hydrophobic is a lot larger than that small hydrophilic region. So these are not readily going to be able to dissolve in water. But because they have that carboxylic acid region, which has a hydroxyl group, it can react with the hydroxyl group on glycerol by removing water, again, dehydration synthesis, and creating what's called an ester bond, E-S-T-E-R. Now, since glycerol has 3 carbons, each with a hydroxyl group attached to it, each of those 3 carbons can have a fatty acid attached to it. So they're typically called triacylglycerols. So we have glycerol, and then we have this triacylglycerol, which forms when glycerol reacts with 3 fatty acids, and each of those fatty acids can be different. So you can see each of the 3 fatty acids can be different, and combining them and taking into account the different combinations of fatty acids that can exist, this creates very, very diverse molecules. So again, the fatty acids can differ, and they can differ in the length. So we can see the fatty acids that's attached to carbon number 2 in glycerol is shorter by 2 carbons than the 2 fatty acids that are attached to carbon number 1 and carbon number 3 within glycerol. And again, they're attached by a ester linkage. So this is an ester bond. An ester bond is formed between a hydroxyl and the hydroxyl group of a carboxylic acid. So we can see part of that carboxylic acid here, and then the oxygen that combines the 2 carbons. This is an ester linkage. So this is characteristic of fats. Now, in terms of the fatty acids, like I said, they can differ in length or the number of carbons within them, but they could also differ in the number and the location of double bonds that can occur between the carbons. So here we see each of these carbons are only involved in a single bond. So we can see that because they're only single bonded to each other, the rest of the bonds are typically hydrogens until the end right here. So we have 3 hydrogens attached to the carbon at the end, and then 2 hydrogens attached to the carbons in the middle because they're all attached to another carbon. Ones that are straight chain. So because the straight chain ones can pack tightly close together, so they're a lot stronger, and heat in terms of room temperature it's not going to be hot enough to melt them. So they're going to be typically solid at they're not solid because they're not tightly packed together, so they can readily move around. Now I want to talk about saturated fatty acids versus unsaturated fatty acids versus trans fatty acids. So saturated fatty acids, again, are going to have the maximum number of hydrogens, so that means that there are no double bonds between the carbons. Now, cis and trans double bonds can matter, right? So in unsaturated fatty acids like linoleic acid, which is found in vegetable oil, it has these cis double bonds between the carbons. And what that means, as I described already, is that the hydrogen that is attached to each of these carbons that are adjacent to one another and involved in a double bond are both on the same side. And again, this creates that kink in the chain. For trans, there's not as much of a kink. They're almost as straight as the saturated fatty acids. Now, trans means that the hydrogens that are attached to the carbons that are adjacent to one another and involved in a double bond are on opposite sides. So this doesn't create really a kink in the fatty acid, and these can still tightly pack at room temperature. So this is going to be found in some margarines, not butter, but some of the margarines, and trans double bonds can occur during the process of dehydrogenation. Now, the function of fats are mainly energy storage, and in animals it's stored in the adipose tissue, which is our fat tissue, and fats provide a lot more energy than carbohydrates. Fats can be unhealthy too. So if you eat a lot of saturated fats and cholesterol, it can cause atherosclerosis, which causes hardening of the arteries or blockages within the arteries and can be fatal. But fats are necessary in our bodies. Even cholesterol is actually necessary. When we talk about the structure of the plasma membrane, we're going to see how important cholesterol is. So eating fats is healthy, but you got to eat good fats, right? Not the bad fats, or not too many of them. So fats are a great source of energy. They also cushion vital organs, and they insulate the body when the temperature is very cold. So here we can see we have adipose cells, and they're basically whitish in color because they contain mainly fat. So phospholipids is the next class. So we talked about fats already. We talked about the formation of fats. Phospholipids are very similar to fats or triacylglycerols, except that, well, they're similar in that they have glycerol as a backbone, and then they have fatty acids attached to glycerol. But in phospholipids, remember glycerol is a 3-carbon molecule which has 3 hydroxyl groups attached. Well, 2 of the hydroxyl groups can react with fatty acids, but the third one is going to have a phosphate attached to it. That's why it's called a phospholipid. So it has a phosphate and then another very large complex functional group which is typically charged or at least hydrophilic. So here we have choline, and this is positively charged because it has that nitrogen there that's positively charged. And we can see that this is the hydrophilic portion. So we have glycerol with the oxygens and the ester bonds, right? So this is hydrophilic as well. And then of course the phosphate and this charged choline will create more hydrophilic properties for, and it's often called the hydrophilic head. But then we have the hydrophobic tails, which are made up of the fatty acids that are also attached to glycerol. So we have this this molecule that's actually, there's a number of terms for amphipathic or amphiphilic, and what that means is that it has hydrophilic properties and hydrophobic properties. Now these are important because these are what make up the plasma membrane. And the plasma membrane is basically the separation. It separates the inside of the cell versus the environment and or the outside of the cell. So it's very important that they interact with each other. And phospholipids, when they're added to water, they can basically self-assemble. So they just start self-assembling into this bilayer or double-layered sheet. So here we can see the 2 layers, and these, of course, these orangish yellow regions represent the hydrophobic tails, and then we have the hydrophilic head, which is an oval grayish shape. Now, these come together because hydrophobic tails like to interact with each other. They're hydrophobic and they like to stay away from water. So they're staying away from water by creating this bilayer and minimizing the hydrophobic tails, minimizing their interaction with water. So the water is going to be on the outside right here where the bluish color is, and the water is going to be associated with the hydrophilic head. So you can see they just kind of readily form these types of bilayers, and sometimes they could create spherical structures, which we'll see some examples that are inside of a cell. But they create these spherical structures, and in commercial products like soaps, they create micelles, and they can create a micelle around a greasy type substances. Now, steroids are lipids that are characterized by this special skeleton, right? So it's a carbon skeleton, and the skeleton consists of 4 fused rings. So we can have we can see here that there are 3 6-membered rings, 6 carbon rings, and then 1 5-membered ring right here. So this ring structure right here is characteristic of cholesterol as well as other steroids. So this picture right here represents cholesterol, and because each of them have this very large hydrophobic ring system, fused ring system, they're very similar to each other. However, the differences are mainly in the functional groups that are attached to them. So this is cholesterol. We can see there's a hydroxyl group here. So you may notice that OH is hydroxyl group, but it could be listed like this, HO, because the oxygen is going to be more close to the carbon ring structure, and then the hydrogen will be kind of hanging off the oxygen. So it's still a hydroxyl group, whether it's a HO or an OH. And then we have a methyl group here and another methyl group here. And then we have this very large alkyl group. So this is basically a very large hydrocarbon. So again, it's only carbons and hydrogens attached to this part. Now let's look at this stick model of cholesterol real quick. Now, let's look at this table right here. We have the components of all the different lipids. We have the fats, which are made up of glycerol and 3 fatty acids. These are called triacylglycerols, so this includes the fats and the oils. And these are important energy sources, so these fats are important, again, in moderation. The phospholipids have this head with the phosphate. Again, they're made up of glycerol, 2 fatty acids, and then we have this phosphate head, which has phosphate group, functional group, plus another charged group that's attached to it. So this makes up lipid bilayers of membranes and membranous structures that are found in eukaryotic cells and also some prokaryotic cells. So again, the steroids have this characteristic fused ring backbone. Some examples of steroids would be, of course, cholesterol, and then we have testosterone and estradiol, which are steroid hormones found in humans and other organisms as well. And what makes them different is the functional groups that are attached to this base backbone. Again, these are important, and especially cholesterol, it's an important part of the cell membrane, which we'll talk about when we get to chapter 7. And they are important signaling molecules, so they serve as hormones. Even cholesterol is a signaling molecule, so it can serve as a hormone, as well as other typical hormones that you might hear of, like testosterone. All right. Nucleic acids. So the role of nucleic acids is really information storage or movement of information in some way, and I'll describe that a little bit later in a little bit more detail later on in the semester. But there's 2 types of nucleic acids. There's deoxyribonucleic acid, or DNA, and ribonucleic acid, RNA. And there's many different types of RNA. Each type of RNA has a different type of function. So DNA and RNA are polymers, and the polymers are made up of monomers called nucleic acids. So they could be called a polynucleotide. So the monomers are the nucleotides. They are connected by what's called a phospho. Nucleic acids store, transmit, and help express hereditary information. Now, remember, you were introduced to gene expression in chapter 1. We're going to go over a little bit more in detail at the end of the semester. However, it's important to talk about now how nucleic acids can store and transmit, and then, of course, help express hereditary information. Now, DNA is going to provide the directions for its own replication. So it stores that information. It stores the instructions for its own replication, which is really cool. DNA directs the synthesis of messenger RNA, so that's a type of RNA, one of the many types of RNA. And it's through messenger RNA, or mRNA, which actually eventually controls protein synthesis. So remember that process that we kind of discussed from chapter 1. We have DNA, which is used to make the RNA or messenger RNA, and then the messenger RNA stores the code that controls protein synthesis. So this creates proteins, which are basically the product of genes. So this process, again, is called gene expression. So we have DNA to RNA to protein, which is the central. Now let's look at this process. In a eukaryotic cell, which happens to have a nucleus, inside the eukaryotic cell we have DNA. Now DNA can synthesize itself, and it stores this information in order to do so. Now DNA also stores information for genes to produce the products of genes, right? So DNA is used to synthesize messenger RNA. Then the messenger RNA is going to eventually leave the nucleus once it's created, and then to the cytoplasm. And the cytoplasm, the ribosome will bind to it and then start synthesizing proteins. So DNA stores the information to actually replicate itself, and it stores the information which is needed to ultimately make proteins through the production of messenger RNA. Now, looking at the structure of DNA, we have what's called a 5' end and a 3' end. The 5' end really refers to that number 5 carbon that's found on the ribose portion of the nucleic acids. So if we look at a nucleic acid, and this kind of points to one of them right here, a nucleic acid or a nucleotide is going to have the nucleotide, which are the monomers of nucleic acids, are going to have what's called a nitrogenous base, a pentose sugar, and pentose means that there's 5 members within the ring, and a phosphate, one or more phosphates, so there could be more than one phosphate group. So this makes up a nucleotide, and a nucleotide can connect to each other through the phosphate group and the 3' carbon to create a phosphodiester bond. So this is a phosphodiester bond right here, phosphodiester bond, phosphodiester bond. And the 5' end of the whole string is where the 5' carbon is going to be, and the 3' end of the string is going to be where the 3' end is going to be. And that 3' end is pretty important for the synthesis of polymers because it needs to be there and it needs to have this hydroxyl group there. So this hydroxyl group will attach to another nucleotide at the phosphate to create another phosphodiester bond when another nucleotide is added. Now, let's look at the nitrogenous bases. There's different nitrogenous bases, and they're called nitrogenous bases because they have nitrogen, and the nitrogen is part of the ring system, and they differ mainly due to either the number of rings that they have, and of course functional groups attached to the ring system. So we have pyrimidines. So pyrimidines are a class of nitrogenous bases that have one ring. So they have one ring system, and there are 3 different pyrimidines. We have cytosine, thymine, and uracil. So cytosine is going to be found in DNA and RNA. However, thymine is only found in DNA, and uracil is only found in RNA. And then we have the purines. The purines are the nitrogenous bases that have 2 rings that make them up. And again, they differ by their functional groups. The 2 purines are adenine and guanine, and those are found in both DNA and RNA. Now, if we look at the sugars and the difference between deoxyribose in DNA and ribose in RNA, the sugars are almost exactly the same, except at the 2' or the number 2 carbon. In DNA, there's no oxygen there. It's just a hydrogen, or 2 hydrogens attached to that one. In RNA, there's a hydrogen and a hydroxyl group. So this is why DNA is called deoxyribose, right? It's missing that oxygen. And ribose is not called deoxyribose because it does have that oxygen right there. Now, if we look at this reaction and how a phosphodiester bond is formed, remember I told you on the 3' end we have that hydroxyl group, which is attached to a carbon which is subject to attack. So you can see that this oxygen as part of the phosphate group in another nucleotide is going to become negatively charged, and it's going to attack that carbon right there. And once that happens, a condensation type of thing reaction occurs, and what this is dehydration synthesis. So water is removed and then the bond is formed, and this happens to be called a phosphodiester bond because of course we have the diester. Remember the ester bonds in the fats? Well, this is an ester bond. We have carbon, oxygen, but it involves a phosphate. So it's called a phosphodiester because there's 2 ester bonds on either side of the phosphate. And these are going to be found in nucleic acids. Now, if we look at the overall structure and the difference between DNA and RNA, so you can see DNA is double-stranded, and it's anti-parallel. And what that means is that one strand goes from the 5' to the 3' direction, and the other strand is attached by hydrogen bonds through the nitrogenous bases in the opposite direction, so it goes 3 to 5. So this shows a little bit close-up of the hydrogen bonds that occur between the nitrogenous bases. Now this suggests that there's only one hydrogen bond that occurs between them. However, that is not true. Between the cytosine and the guanine, there's 3 hydrogen bonds, and between adenine and thymine, there's 2 hydrogen bonds. However, it just shows that this is just to indicate that there are hydrogen bonding that occurs between the 2 strands of DNA, and it creates this helical structure. So we have this helical structure in which you have a major and minor groove. Now, RNA can take on a number of different overall three-dimensional structures. RNA is only a single strand, but you can see it's kind of folded upon itself, and it creates this overall 3D type of structure. And here this just shows the ribbon diagram, and you can see, again, hydrogen bonding can occur between the bases. However, it's between bases within the same strand, not between strands. So this happens to be an example of a transfer RNA, in which we'll learn a little bit more about that later. It's not important what the function of transfer RNA is. So far you've heard of a messenger RNA, and now you've heard of a transfer RNA. There's other types of RNA, and they each have their own type of function. Now let's take a look at the stick model of DNA. Let's see if we can get this working. All right. So we can see the overall structure of DNA, and you can see the major and minor groove, and you can see the nitrogenous bases, which are going to be oriented about horizontal to each other if you have the DNA standing up on its end. All right, so remember, as a summary, we have the components of nucleic acids, or nucleotides, which make up nucleic acids. So a nucleotide has nitrogenous base, and again, it could have 1 or 2 rings. This is just a symbol for the nitrogenous base. It doesn't represent its actual structure. It can have a pentose sugar and 1 or more phosphate groups. So the examples of nucleic acids include DNA and RNA. Remember, there's different types of RNA, and the difference between DNA and RNA are the following. DNA is double-stranded, and the strands are anti-parallel. The sugar is deoxyribose. Remember, it's missing the oxygen at carbon number 2. The nitrogenous bases within DNA are C, G, A, and T, or cytosine, guanine, adenine, and thymine. And again, it's usually double-stranded. RNA is typically single-stranded, and for purposes of this class, let's just say it's single-stranded. The sugar is ribose, not deoxyribose, so the ribose has that hydroxyl group and a hydrogen at carbon number 2. And the nitrogenous bases found in RNA are cytosine, guanine, adenine, and uracil. So you can see that last one is different. Remember, in DNA, thymine is present, and RNA, uracil is present. So DNA stores hereditary information. However, RNA has many different functions. Most of them have some function in gene expression and including protein. All right, so let's talk about genomics and proteomics. We haven't really talked about the structure of proteins, but in terms of DNA, which makes up the genome of an organism, the structure of DNA and its relationship to the amino acid sequence is quite important, which allows us to decode genes. And we can decode genes by determining what the base sequences of genes are. So back in the 1970s is really where scientists started sequencing DNA, which is basically determining the order in which the nucleotides are connected together within the chromosomes. Now we have better methods to sequence DNA. Before we could only do like little small fragments and then try to put them together. Now we can sequence over 200 whole human genomes in a day. Now we have a lot more higher technology methods now. This kind of brought up the Human Genome Project, and then of course since then many genome sequences of many different organisms have been determined. Now, bioinformatics is basically the area of science that deals with the use of computer software and other tools in order to analyze DNA sequences. Bioinformatics pulls these DNA sequences from databases and uses computer software to compare the DNA sequences and the protein sequences. So this kind of brings us to this area of science called genomics. and proteomics. So genomics studies the genome sequence of organisms, and proteomics is the area which studies the protein sequences and their functions within different organisms. So the sequences of the genes and their protein products can be used to document the background, the genetic relationships of organisms, hereditary background of a particular species, like create a pedigree. And then you can determine how DNA is passed from offspring to, or from the parents to the offspring. We can extend this concept of molecular genealogy to relationships between species. Can determine, how species have changed over time, or study the evolution. We can study fossils, paleontology, identify fossils, and then study their relationships between other fossils and the time in which they existed. We could use it in medical science and, of course, conservation biology, and kind of analyze the interactions between species within the ecological systems. A lot of information that we can determine from protein sequences and DNA sequences. So in summary, we talked about the structure and function of lipids and nucleic acids, and talked a little bit about the areas of science dealing with nucleic acids and proteins, and what information we can extract from these sequences. Well, thank you for listening.